we're gonna finish up chapter 14, which is dealing with our blood vessels and then also how our body regulates blood pressure. And we left off our discussion yesterday with understanding how the blood pressure is monitored and regulated in our body. And we learned, just like what we've been putting together all semester, this as well, controlled extrinsically by the nervous system for precise, quick regulation, and then more long-term can be controlled utilizing the endocrine system targeting the kidneys, right, to alter blood volume. So let's just very quickly review, because we took a little bit of time yesterday to talk about how the nervous system controls our blood pressure, which nervous system regulates our blood pressure. Very good. Autonomic nervous system. So the effector organs which maintain our blood pressure are duly innervated by the sympathetic and somewhat the parasympathetic nervous system. The receptors that monitor our pressure are called. There are some chemoreceptors. There's also baroreceptors. Okay, so both are involved. One monitoring chemicals, the other monitoring specifically the pressure inside of the vessels. They'll communicate afferently to cells in our what? Medulla oblongata, which is the control center in this pathway, as well as a lot of our other kind of basic survival pathways. When out of balance, either way, the efferent control will target which effector organs. What are the effector organs in this pathway? The heart and our blood vessels. At the heart, we can change the what? Two things. We can change the pace, and what'd you say? I wouldn't use that word diameter for the heart, but definitely we could talk about contractility, right? So we can change the contractility of the heart and the pace of the heart, which we know basically changes the what? Cardiac output. Also, though, we learned that the vessels are targeted, so the effect there is to change what? The diameters, which influences what about a vessel? Its resistance. And then obviously, if your blood pressure is beginning to fall below set point, which nervous system will become activated? Which subdivision? When you're falling too low, sympathetic nervous system needs to be activated. So your heart rate goes up, contractility goes up, resistance goes up, and hopefully pressure also goes up. Okay, so it's just this negative feedback balance to maintain your pressure in that set point range. Textbook will tell you 120 over 80 systolic over diastolic. Okay. So coming together a little bit now with hopefully the nervous system, the role of that familiar receptor, control center, effector, negative feedback, and repetition we've been seeing all semester. What questions do you have about putting that into practice here at the heart, cardiovascular system? Where we left off on yesterday was adding a little bit more complicated, if you will, or more detailed process to monitor blood pressure when the blood pressure is out of set point for more than a few milliseconds. So if we're looking at a more prolonged blood pressure change, the endocrine system can get involved by acting at the kidneys. Okay. And how the kidneys can help resolve any pressure issues is actually just changing the volume of blood. Okay. If our blood pressure is too low, the kidney needs to do what? The hormones will be involved, yes, and the net effect is going to be what? Increased blood volume. Okay. How could the kidney increase blood volume? Let's not even get specific. Yeah. The kidney can increase your blood volume by not doing what? Not making as much urine. Okay. And so it takes a bit. It is going to involve those two hormones which are just mentioned, renin and aldosterone. Antidiuretic hormone will also be involved. Okay, so you guys are Right. So if I have a long-term low blood pressure, my kidneys, they'll still filter blood, but they'll, what process at the capillaries? They will reabsorb a high proportion of it. So our urine output is less. Okay. But if I have high blood pressure, okay, if I've got familial history, my kidneys are going to do what? Filter. Will they reabsorb as much? So my urine output will go up, hopefully to bring my blood volume down, to bring my blood pressure down, hopefully to set points. Okay, and that's what we're going to look at, this specific kind of pathway. The endocrine system's involved, the kidneys involved to change our blood volume. And so we're going to look at two pathways, one considered to be the direct mechanism at the kidney, and the other one's going to be indirect. And so we left off with this image that you have there in your guided notes. And we labeled the direct pathway on the far left end of this image. And what we're going to learn in the last probably day of the class, actually, is the kidneys themselves are mechanically sensitive to the flow of blood. Okay? So if a lot of blood is coming to the kidney, is reading that as high what? High pressure, right? Or if there's not a lot of blood going to the kidney, it's going to decide that as low pressure. And so directly the kidney, right, can detect this. Right? So just physically detecting the blood flow is an ability of the kidney. So if low blood flow to the kidney, you're not going to filter. You're not going to make urine, so effectively the blood volume should stay up, which should bring your blood pressure back up. Okay? So where we're going is the kidney can do this itself, okay, which we'll repeat next week. Just trying to understand once we look at the structure of the kidney. Does that make sense? You can directly determine the blood going through it. Although it looks kind of crazy in this image, is actually a review from the endocrine system for you, and how the hormones can target the kidney to alter our blood volume and ultimately our urine output. So if for some reason your blood pressure is low over an extended period of time, and the nervous system, by changing resistance and output, is not fixing it, the kidneys can be targeted hormonally. And so we have this indirect pathway. Okay, so we have baroreceptors. Sympathetic nervous system is actually going to stimulate the kidneys to release this product called renin. I'm kind of mentioning it. I mention it now, and hopefully next Wednesday we'll get to better flesh it out. But what it does comes from the kidney, and it targets the release of something called angiotensin 2. Okay, so we're just going to look at this, then this, then this. Okay. Angiotensin 2 is probably new to you, but what it stimulates is not new to you. It stimulates the release of aldosterone from where? The adrenal cortex. What was the effect of aldosterone? Targets the kidney to not it does from the cortex, but not this different hormone. Reabsorption of a particular ion, sodium. Okay. When aldosterone targets the kidney tubules, sodium is reabsorbed back into the blood. Sodium's a charged particle, and it actually creates an osmotic gradient. So go back to that second lab experiment we did. Remember, we put our cells in the hypertonic solution. The cells began to sodium was out and drew out the water. So it's kind of envision the cell like what would be your urine. What would be urine goes back into the blood. Okay, so it creates an osmotic gradient. Sodium is reabsorbed, and then so is water. Water goes back into your blood, so your blood volume goes up. Okay, to hopefully bring your blood pressure back up to set point. Okay. So renin cascade triggers an intermediate, causes aldosterone, which promotes directly sodium reabsorption and indirectly water reabsorption. As well, it causes the release of, what is that? Antidiuretic hormone. It directly targets the kidney to reabsorb what? Water, directly. Okay? So taking it out of the filtrate, or what would be maybe urine, putting it back where? Back. Putting it back into the blood. Okay, whereas the kidneys, we're taking it from the filtrate, putting it back into the blood. So our blood volume goes up, and hopefully our blood pressure goes up. Okay? And that is the indirect pathway in a crash course, if you will. But hopefully a review to the endocrine system about aldosterone and antidiuretic hormone. You can also see another effect of angiotensin II. What else does it target? Blood vessels, probably to do what in low blood pressure scenario?
Increase the blood pressure.
Yes. How?
Decreasing
By constriction, right? So increasing resistance is the term that we've been using. But yeah, decreasing the diameter, or we could say increasing resistance, which effectively brings our blood pressure back up to set point. At this point, I'm hoping that you can appreciate how our nervous system can quickly target the heart and our blood vessels to have an effect, to maintain blood pressure. But if a blood pressure problem, if you will, prevails, how the endocrine system can get involved to change our blood volume. What questions do you have about that relationship of how the nervous system, endocrine system can regulate one's blood pressure? Okay. Going once. Chapter 15 coming up then. Chapter 15 is going to discuss our blood, and it's going to get into all of the different blood cell types. And honestly, we don't have time to go into the different blood cell types. So I'm going to focus predominantly on one blood cell type. Which one? The red blood cells. Okay. So when you go on to you take immunology or microbiology, you'll get into better detail of the different types of white blood cells. Today, we're just going to say there's white blood cells out there in the body, and kind of briefly talk about what they do in general. Okay. If you were to look at blood on a light microscope, or just we call it a blood smear, the bulk of your blood is actually the red blood cells, which you guys just mentioned. And a smaller proportion, and a healthful individual, should be our white blood cells. And you may also see some tiny little blood platelets, these little fragments of cells. When you go on to take your immunology class or microbiology, what's going to differentiate the different types of white blood cells which we have is the shape of the nucleus. They have really cool and different shaped nuclei. So that's how you can determine which one is and what it does, whether it's designed more to defend against bacteria or viral or whatever the case may be. But most notably, what is noticeable characteristic about red blood cells that we can see about in this image? What can you tell about red blood cells here? Okay. What is their shape? They are biconcave. Very good. What else can we tell about red blood cells from this image? There's a whole heck of a lot of them. Very good. What else can you tell about red blood cells from this image? No nuclei, right? Can you see that? We can see the nuclei in the red blood cell, or assume the white blood cells, but noticeably absent in the red blood cells. If you look at a given cell, it looks really kind of transparent. Okay? And we'll talk about today why that is, and why there is no nucleus inside of our red blood cells. There's some really neat scanning images of red blood cells. These, like the other scanning images which we've used all semester, are considered to be false colored, so colored post-data acquisition, not grayscale. but the red blood cells have been colored red so we can see them better in the image, and the white blood cells left in the grayscale. But you can see the lumen of a vessel. You can see some of the musculature. You can actually see the capillary to the vessel, and illustrating that the vessel's an organ too. It needs its own little blood supply. But I like this image for multiple reasons, including, can you notice how the white blood cell actually adheres to the vessel? You may recall from our discussion about vessels how I mentioned white blood cells can actually get through the little capillaries. It's because they have those extracellular proteins that they use to kind of motor around and move about the body. We can also see through some of these cells as well that biconcave shape of the red blood cells. Some of the cells look really odd and misshapen. I'm presuming that's probably from just collecting the image. If you prepare any tissues, you have to, for microscopy, you have to dehydrate them. And so you're going to change the shape of them. So I'm going to guess these cells are not diseased, and probably just from the taking the image, basically. Okay. So yeah, red blood cells are really unique because they have a really unique function inside of our body. A very quick video, and this is, I think, the video that I asked if I showed, and someone said, "Oh yes, you did," and y'all are like, "I don't think so. I don't think I showed it to you guys yet." So I have some samples of some vessels.
You might have heard the saying that blood is thicker than water. Well, the saying is true. That's because our blood is packed with millions of cells that float in a river of plasma. Most of these are red blood cells. They're one of the few types of cells without a nucleus or mitochondria. These flexible discs can bend and squeeze through the smallest capillaries of our body. They get their red color from hemoglobin, a protein that lets them transport oxygen throughout the body. This process keeps us alive. When blood passes through the lungs, millions of red blood cells pick up oxygen for transport to other cells in our body. When we exercise, for instance, our cells need more oxygen. Our lungs breathe in more air, and our heart pumps faster to speed up the delivery of oxygen-rich blood. Red blood cells also act as garbage collectors. They remove carbon dioxide by carrying it back to the lungs. Every day we replace millions of these cells. Each lasts around 120 days, but our bone marrow continually replenishes the supply. Our blood also holds an army of white blood cells that spring into action upon infection. They defend our body against illness and disease. When an infectious agent enters the body, these cells rush to attack it. What makes them so effective is their ability to change their shape. They squeeze through the walls of blood vessels to battle the enemy head-on. These bacteria cells don't stand a chance. White blood cells surround and devour them with ease. In the case of troublesome viruses, white blood cells produce antibodies that mark them for destruction. We also have other components in our blood that help control bleeding. Platelets unite to plug up holes in our blood vessels and stop them from leaking. Without them, we would bleed to death from the tiniest cut. The clots formed from platelets also keep harmful germs out. The cells in our blood perform vital functions in our body, delivering essential materials, removing wastes, and working together to keep us healthy.
All right, lights on. Were you able to see those vessels dilating and constricting in the image? And how the capillaries, like, they were just letting one little red blood cell through at a time? Hopefully that was kind of a bit of a nice visual review from which we've kind of talked about for the different vessels so far. We became familiar with the blood early in the semester, and primarily in which we described it for a component of the extracellular fluid. We talked about the blood plasma, okay? And it is, with the exception of our lymph, which actually goes eventually into our circulatory system, it's only liquid or fluid tissue. And it does have the liquid plasma, our blood plasma, which we've introduced early in the semester. There's cells in there as well. And the cells which you find inside of the blood include your erythrocytes, your leukocytes, and then platelets. I'd like for you to identify on your guided notes that your platelets are not whole cells. They're actually pieces of a whole cell. They're considered to be cell fragments. Erythrocytes is the proper term for our red blood cells, and leukocytes is the proper term for the white blood cells. And I tend to just use the abbreviation RBC for red blood cell and WBC for a white blood cell. Just like everything else inside of our body, there's a set point range for the proportion of the plasma to the formed elements. And in order to determine the right proportion of red blood cells, you would have to measure one's hematocrit. And so the hematocrit is just a term to illustrate what percentage of a volume of blood is actually red blood cells versus the plasma versus the white blood cells versus the platelets themselves. Some of you guys may actually work in the lab. Does anybody do this? Just spinning out samples, getting anybody out of this lab? Sometimes there's somebody that gets to work in a lab and collect and measure. So if we were to look at a hematocrit, like you can see there in your handout, in order to do so, obviously you would need to take a sample of blood. You'd centrifuge it, which just high velocity spins the sample and it separates the more dense versus the least dense substances. Okay, so red blood cells are clearly going to settle at the bottom because they're packed with hemoglobin, like this big, huge protein that has an iron, like a metal inside of it. So it's really heavy and dense. And then above it would be considered what's called the buffy coat. And in the buffy coat, you'll find your white blood cells and your blood platelets. And you'll notice it should be pretty minimal. And the balance of your blood cell would actually be the liquid plasma. And it does have this kind of golden appearance, which you can see in this image. So it's sometimes described as like straw-colored. It's like kind of like your urine, right? Because your urine is just filtered blood plasma. Okay, so it has that kind of yellow color to it. So if you were to look at a hematocrit, about 45% of your blood should be your red blood cells. So about 45 is a good hematocrit, kind of the textbook kind of set point published number. A higher proportion, about 55% of your blood should be plasma, and the minimal amount of your blood should actually be white blood cells and platelets. Obviously, if those ratios change, it indicates hydration or infection or some other blood condition or whatever. So just looking at the composition of the blood can dictate one's health. Or not dictate it, but indicate if someone's doing okay or not. Okay. What questions do you have about using the word hematocrit? What's in the whole blood? All right. If we look at blood, it's always red. It's never what? Our blood is never what color? What we see in our books. What color is our blood not? That you see in books. It's not blue. Why do we see our veins and stuff colored blue then? I gotcha. She said to illustrate a vessel which contains blood that has less oxygen, okay? And that's all it is. Okay? Your blood should not be blue because the metal in our blood is which metal? Iron. And we know here in Oklahoma, thanks to the ground, right? Iron turns things red. Okay, so your blood is red. The degree of redness depends on the relative amount of oxygen bound to the hemoglobin in your red blood cells. And so the more oxygen which is found in the blood, the more bright and brilliant it would be. And the less oxygen which it has, the more dense and dull kind of appearing, if you will. Okay, so it's just a range. If you pull out venous blood, it'll be really dark. We can almost use the jackets in the room, Mashihidi, your sweater. Okay, I would guess maybe if I were to do a visual, okay, between you two ladies, venous blood, arterial blood. Okay, yours are bright red. Can y'all see the visual? So that's kind of the difference. But your blood should never be blue, unless you're like a horseshoe crab or something like that. Other animals do have blue blood, but we don't. We have iron in our blood cells. The range of blood for pH is 7.35 to 7.45, and that'll tie right into the respiratory system lab, which we are doing today and yesterday. Okay. And so the respiratory system is certainly going to work with the cardiovascular system to make sure that blood pH is within that set point range. If we skew off of that a little bit, the body's out of homeostasis and something probably is respiratory, right? Probably a respiratory problem going on. Okay. The blood temperature is a little bit warmer than the core because the blood is distributing not only just nutrients and hormones, etc., but it's also distributing temperature. Right? So when you draw blood, the vial is like really warm because blood is warmer than your core temperature. About 8% of your total weight. Right? So when you hop on the scale and you feel sad, you're like, oh, well, that's blood plasma, right? And all of that iron in there. Iron is heavy. So a lot of the overnight weight loss pills, they're like, oh, you can shed 20 pounds overnight. What are you going to lose? Blood volume. Okay. Hopefully not the whole red blood cells, but you're actually going to lose some blood plasma or something like that. You're going to become what? You lose 20 pounds overnight. Severely dehydrated. So don't do that. Okay, not a good idea. Okay. So it does have a high proportion of your total weight in your body. The volume is obviously proportionate to your size. Since males tend to be larger than females, males tend to have more volume of blood, and ladies tend to have less volume. So we say 4 to 6 liters is pretty average volume for blood. And we guesstimated it last week or a week and a half ago when we did our cardiac output at rest. Gives you a good idea of how much blood you actually have in your body. If we look at the blood, it's going to distribute, it's going to regulate humoral substances like we saw with the endocrine system, and it does give protective mechanism as well. So a couple of these 2 points, 2 of the 3 points are going to be familiar, and then the third point, protection, we'll kind of briefly talk about but not give really the justice which we need. Some examples of substances which need to be distributed thanks to the blood, and again, kind of repetition throughout the theme of the semester. We know distributing oxygen right from the lungs to the tissues, likewise nutrients from the digestive system to our systemic body, picking up waste, picking up hormones, picking up neurohormones, whatever, you name it. The circulatory system in the blood can carry it from point A to point B. If the thing is small enough to get into the blood and is soluble, or if it needs a carrier, right, all those factors tie right back in there. So I think of the 3 things, you're probably most familiar with that. The blood distributes. It's part of the circulatory system. It also regulates things which we become familiar with. Obviously, our body temperature is certainly regulated. Where the blood goes, that area is going to be warmer. Where there's less blood, a particular part of the body is going to be cooler. Because we have a set point range of pH in our blood, that extracellular pH is going to influence intracellular pH. So it's going to directly influence the health of tissues and the pH of those tissues as well. And obviously keeping the fluid, what does it stand for? Volume, very adequate. If our extracellular fluid is good, our interstitial fluid is good, interstitial fluid should be fine, right? So it directly influences the health and the fluid volume and molecular component intracellularly as well. So we're distributing, we're regulating, but also protecting the body. this is the portion of the semester which I mentioned way back in, when we started in June. So I'm going to talk about the immune system for about this long, but really we could talk about it for a good semester or longer. So I didn't want you to equate this little bit of a discussion as to me not appreciating the immune system. We just don't have time. Okay, so we'll just kind of look at a snapshot of how the blood system gives us protection. And two primary ways that the blood can give us protection is by physically preventing blood loss, which you're probably familiar, by utilizing the formation of blood clots. Blood clots are formed in order to prevent, if there's been a blood vessel damaged, it's been cut, a clot can form to kind of seal up, plug up that hole, if you will, to prevent any excess blood being lost so you don't kind of bleed out and die. Okay? The way it works is by utilizing proteins that are already in your blood plasma and platelets, those little cell fragments that are in your blood. They're called to action, if you will, once a tear is acknowledged inside of a blood vessel. So they can, it's a very intricate messaging pathway. It's like, and I think your book goes into the detail, but I'm not going to hold you accountable. It's very intricate. But our good friend calcium's involved, helps initiate that clot formation pathway. But you'll notice we can begin to appreciate, thanks to figure 15.7, the role of fibrous types of proteins and how they can actually link and tie two different regions of a damaged area together, and they contract. And so almost think almost like a bridge. Okay. Instead of being a waterway below the thing in which the bridge is going over would be a wound. It's got an open wound, and the platelets will lay down proteins like a bridge in that analogy, and then those proteins will pull together, right, just bringing the pieces, the wound, right, basically together to allow for it to heal. And so it's a very intricate process to lay down those extracellular proteins and allow for them to be active and plug up the kind of cement the hole. So our platelets and proteins inside of our blood plasma facilitate that when there's damage. Secondarily, our blood gives us immune function by preventing different types of infection, whether they be viral, fungal, bacterial, whatever the case may be. We have different blood cells to protect us against different types of potential pathogens. And we also have the ability to synthesize things called antibodies. You've heard of those? What's an antibody? What is an antibody? Heard of it. What is it? Anybody have an idea? What is an antibody used for? Very good. It's to fight infection. And basically it's a molecule that's produced to recognize pathogens which you've already been exposed to. Okay, so for example, if you get the flu vaccination in the fall or spring, whenever you go get it, right, exposed to that vaccination, right, they're basically giving you what? When they give you the vaccination, they're giving you the virus, and it's considered to be killed. It's not really ever living, but it's inactivated. Okay. You're exposed to the pathogen. Your body says not good, so it starts producing antibodies, those little molecules that stay in your blood. And once they see that pathogen again, they can recognize it like a receptor, basically, and then call in new white blood cells to just degrade it, metabolize it. So the ability to make antibodies. Other white blood cells can actually phagocytose bacteria and fungi and all kinds of good things. So blood is good for keeping us kind of healthy, if you will. That is like as simple as I can get on the immune system. Okay. Do you guys have any questions about platelet formation or just in general infection, antibodies, platelets? Yeah, it's got you slowing down. All right. So let's look at more specifically the red blood cells, or the erythrocytes. As described a moment ago, they are biconcave. We worked through the phrase bi means two. Concave means which direction? Inwards, right? And so the red blood cells have a biconcave shape, and they're considered to be discs, so they are round, and they are anucleated. We learned already means without a nucleus, and that's in their mature form, and so I'll clarify that coming up. And they basically don't have any organelles in the mature form either. Okay, so they're pretty stripped down, if you will, compared to other cells which we've studied so far. They're filled with a specialized protein called hemoglobin. You can abbreviate that HB. It's not the only protein in there. If you were in lab yesterday, what other protein would you find in your red blood cells? Carbon dioxide is in there, but it's not a protein. That's just a gas. What other protein did you become familiar with inside of red blood cells if you were in lab yesterday? Carbon dioxide can go in there and interact with this protein called bicarbonate ion. Is an ion, so it's not a protein. But this thing helps form that, called, yeah, I wrote it on the board, you wrote it in your handout. Something like that. Carbonic anhydrase. Very good. So another example of a protein which you would find inside your red blood cells is an enzyme called carbonic anhydrase, which we'll talk about in lab today, or you spoke about yesterday. So it's not just, hemoglobin is not the only protein which you would find inside of a red blood cell, is where I was going. But it's like crazy packed with hemoglobin, it's just jam-packed with hemoglobin. And the primary job for hemoglobin is transporting gases such as what? Carbon dioxide and which? Oxygen, right? So those are the two primary gases which it will transport. Very good. In the membrane, it's a plasma membrane. It also has a protein in high abundance in this membrane called spectrin. And spectrin allows for those red blood cells to squeeze between those small diameter lumen capillaries and jam through, just like we saw in the images, right? We don't want them to be hard and rigid. If they're hard and rigid, they probably get caught in our different vessels. So they've got to be a bit flexible to kind of make their way through the blood vessels. And so their lipid, I can't remember the exact composition, but the lipid content of the red blood cells is a little bit more specialized to give it this flexibility as well. Here's a couple of images. The image on the left is a scanning image of a red blood cell. Again, it's that false color. And then the image on the right is looking at a cross-section, illustrating that biconcave nature of the red blood cell. So it really looks completely different from any cell which we've studied all semester. But of course, we know that structure dictates function. So it's a different structure because they're a really unique function. And the biconcave shape is really critical in that it helps have a huge surface area, and I want you to clarify this statement. It has a huge surface area in comparison to the cytoplasm. Okay, so it has a lot of surface, but not very much what? Not very much cytoplasm. So what we're adding is erythrocytes, that biconcave shape gives a huge surface area in respect to the cytoplasm. Why do you think we have to have such a big surface area for a blood cell, a red blood cell? We've mentioned surface area some throughout the semester. Why do you think we need a large surface area across a cell that's mobilized? To transport, and you're on the right track. It is transporting. Do what? We did an experiment on this. We talked about, I mentioned it during an experiment. I guess we didn't directly do it. What about surface area? As we increase surface area, what also increases? Did you say volume? Well, we actually have a very small volume inside of this cytoplasm. So it depends on what volume you're speaking of. So let's flesh it out a little more. If I increase surface area, what can I increase across a cell? Transport of where? Kind of. If the surface area is great, yes, we can transport, and more specifically, the type of transport would be diffusion. Greater surface area, rate of simple diffusion goes, if my surface area goes up, my rate of simple diffusion goes up. Because, what do we need to simply diffuse across this membrane really quickly? What do we need to move across the membrane really quickly? Some, yes, but more specifically, first, before we move ions, red blood cells, oxygen and carbon dioxide. We've got to get oxygen in there easy. We've got to get it out of there easy. We've got to get carbon dioxide in there easily. We've got to get it out of there easily. Okay? One way to help that is just have a huge surface area. The greater the surface area, the greater the rate of diffusion of gases into and out of the blood cell. So there's a method behind the design, in that biconcave shape does have a huge surface area in respect to the cytoplasm, so that we can have greater rate of simple diffusion of, say, oxygen, carbon dioxide. But ions too are going to move across the membrane, so we're going to go there in just a little bit. I'm hoping we're getting into the semester. Hopefully we're starting to piece all that general stuff that we learned in Unit 1, starting to put it together in specific cell types here throughout the rest of the semester. So again, we said our red blood cells have a huge surface area in order to enhance that simple diffusion. ATP, it's a living cell, but it's kind of different. It still needs ATP, it still needs to transport, you name it, but it doesn't have a mitochondria, it doesn't have a nucleus. And so it has to generate ATP under anaerobic conditions. Okay, so no mitochondria, no Krebs cycle, no electron transport chain. And the point is highlighted there in your handout. We want our red blood cells to transport oxygen, not do what with it? Don't use it. Okay? And so one way to prevent it from using it is just kick the organelle out, which would utilize it. So not only do our red blood cells not have a nucleus, they also don't have mitochondria within them. We look deep within the red blood cell and looking at a given hemoglobin molecule, it can bind with oxygen reversibly. So what does that mean? You can reversibly bind oxygen. What does that mean? You can bind and unbind, okay? Which makes sense, right? Not only do we pick up oxygen, we want to drop it off, let it go. Let it go. Well, 2-year-old. All right. If we look at the structure of hemoglobin, maybe I'll need to throw that little sound clip in here, don't I? Let it go. If we look at hemoglobin, globin, what type of protein is it? Shape-wise, when we talk about filamentous and globular, it's probably globular, hemoglobin. Okay, it's a globular protein, and it has 1, 2, 3, 4 little subunits to it. So it has 4 little pieces making up this one big protein. Within each of the pieces, or what's called the subunits, each subunit has an iron core called a heme group. Okay? And it's that iron atom that actually is the reactive part that binds to the gases like oxygen. It forms a temporary little electrical attraction between the respiratory gases. Okay? The globin subunits, there are 2 alpha chains, just based on their shape, and 2 are called beta chains. Okay, 1, 2, 3, 4. Therefore, each hemoglobin molecule can carry how many oxygen atoms? 4 per hemoglobin molecule. And the question is, well, then how many hemoglobin are within a given red blood cell? Guesstimate maybe about 250 million hemoglobin molecules per red blood cell. So each red blood cell could carry about how much oxygen under ideal conditions? $250 times 4, about 1 billion molecules, okay, under ideal conditions. Okay. So in other words, each red blood cell has a high oxygen carrying capacity. Here's a couple of images of the red blood cell. We're taking a single red blood cell out, zooming in, finding a single molecule. You can see 1, 2, 3, 4 little globin subunits. And the little yellow disc is illustrating the reactive iron core. And in this image, you can see one oxygen atom bound, and then the others are waiting to be bound. A couple of other images. Figure 15.3 also illustrates the subunit structure of hemoglobin. There's variants of the alpha subunits, the darker red, 1, 2, and the beta subunits in the lighter red. Again, the iron reactive core is shown in the yellow disc. So this is the reactive part where the gases can bind and unbind. We already have an idea, thanks to lab yesterday, or if you're coming today. We know that hemoglobin not only binds to oxygen, but it can bind to other gases, such as carbon dioxide, it can also bind hydrogen ions, and the effect is, right, if it binds hydrogen ions, they're not out in the blood plasma. So it's a way to describe here, buffer the pH, and so it'll make the pH maybe more alkaline if it needs to be. Right? So you can just bind hydrogen ions. It can also bind to carbon monoxide. Heard of that one before? Or is this a big deal? In houses, when do we hear about carbon monoxide the most? Gas furnaces in the winter, and someone has just died. Dun, dun, dun. Right. So, because carbon monoxide can bind to hemoglobin, but it's almost irreversible. Under normal atmospheric pressure, it's considered to be irreversible. So if carbon monoxide is present and it's binding to your hemoglobin, and what? It's not going away. And so effectively, what happens to your body? You're poisoned, and what happens? If the hemoglobin is carrying carbon monoxide, what's it not carrying? Oxygen. So what happens to you? You suffocate and then you die, right? You don't have oxygen to pick up electrons in the mitochondria. And your ATP shuts down, and it's not good. And so if you smell a gas leak in the area, or if you have those quick onsets, like headache or whatever, get out of wherever you're at. And if necessary, if someone's getting nauseated, you go where? Where do you need to go? A fresh area, and who do you need to call? 911. Immediately, because they can take you to the hospital, put you in a high oxygen pressure chamber, force those carbon monoxide molecules out of your hemoglobin, and hopefully save you. Okay? PSA, my public service announcement. It's scary. It's quick. It takes you on before you know it. It can also bind to another gas called nitric oxide. Nitric oxide is a gas which effectively has an effect of vasodilation, and is probably more aware, you're probably more aware of it in the media if you see all those ED ads on TV. You know what I'm talking about? Okay. Vasodilation is what drives that. Okay, so nitric oxide is just a natural vasodilator, and it can be kind of hitch a ride, if you will, with hemoglobin. You may recall we just mentioned that our red blood cells don't have a nucleus, and so the question is, well, if they don't have a nucleus, where the heck do we get new red blood cells from? Okay, and where do we get new red blood cells from? Our bone marrow. Okay, and so we have some cells in our bones, in the marrow, which produce, they're called stem cells. Have you heard of those things before? Or is out in the media, right? Well, we have stem cells in our body, and they're programmed to make our blood cells. And so we always have to make new red blood cells, because our red blood cells themselves, they don't have a nucleus, they can't repair themselves, so they die, right? After a short amount of time. And so the process to manufacture new red blood cells is called erythropoiesis. And it's a process of making new red blood cells. So we have these cells in the marrow. They're just kind of generic looking cells, and they will continually divide to manufacture our different blood cells. The term pluripotent means they're already programmed to be a blood cell. They're not going to randomly turn into a nerve cell or a fat cell or a muscle cell or whatever. They're going to turn into a blood cell. We know that our red blood cell count, just like everything else, should be in set point range. So if I lose red blood cells, I better be making new red blood cells. If I'm making too many, turn it off, right? Set point range for red blood cell production. So it should be in set point range or relatively constant, as illustrated here. The way that your body knows if you need new blood cells is just actually monitoring the oxygen content in the body. If our oxygen levels decline, relatively decline for a period of time, the body will be triggered to undergo this process called erythropoiesis. And the process is controlled by a hormone which has the same prefix, but the suffix tin tells us it's a little protein. The little protein called erythropoietin triggers the process of erythropoiesis. I think I have the abbreviation coming up, but I'm not sure. So if you just want to parentheses this, EPO, erythropoietin. You heard of that one before? You heard of that acronym before? Erythropoietin? Or EPO? May have heard of EPO. You probably, does anybody watch sports? Anybody watch? Yeah. Where I became most familiar with it in the media, and I think a lot of folks did too, was in 2007 during the Tour de France. Remember that? Who was the big guy winning? In 2007, who was doing all the winning? Mr. Armstrong, right? What did he get accused of? And I think finally admitted but didn't admit to. Blood doping, taking drugs, including a mixture of a hormone called erythropoietin, EPO. Why would an athlete want to dope, inject themselves with something called EPO? Effectively, erythropoietin will give them more oxygen. So it'll give them more blood cells and therefore more oxygen carrying capacity. If each blood cell can carry 1 billion molecules of oxygen, what can I do more if I've got more blood cells? Work out longer, aerobic respiration, and longer. So it kind of gives someone an edge. And yeah, they can blood dope as well. Muzzle tongue. You can extract your blood. So you take it out. You take out your blood. What's your body going to do? Make more. Okay. So I put all my little blood cells over in the bank in the refrigerator the night before the big race. Put it right back in. That's not illegal. It's my blood cells, right? What do you think happens to your hematocrit? Sky high, so what should you then do? Drink more water, right? Okay. Bring your blood volume back up. And so your blood pressure goes up. You're going to, right? I don't know. I just don't think it's a good idea. But those athletes do. Okay. If we, again, this process is very much intricate, but we'll kind of get a snapshot of how this happens. If we were to look at the stem cells in your marrow, they look really just stereotypical boring, like we've seen, like when we talk about generic cells, right? They will be programmed to divide. Okay, you know, when cells divide, there's one goes into two, so one stays just there to divide, and the resulting daughter cell can go on for further development. If we look at specifically the development of the erythrocytes, okay, the cell will then have to get its structure right. So it's got to program itself so that it incorporates spectrum and make it flexible. Incorporate carbonic anhydrase so that it can, right, convert carbon dioxide by carbon ion. It's got to definitely put in a ton of hemoglobin, right? Millions of molecules of hemoglobin jammed in there. It's got to get all those molecules built and ready to go. And so that's what it does during this kind of growing phase. And then we said, well, this erythrocyte needs to get tiny so that it can squeeze through our little capillary. So it just effectively reduces its size. Also, mitochondria, nucleus, they get ejected. And then as the hemoglobin becomes functional, that iron core picks up oxygen, and then the red blood cell takes on its characteristic red color. And so it's really neat developmental pathway to produce a functional red blood cell. And the process is called erythropoiesis. Erythropoietin, EPO, is going to release when our relative oxygen levels are low. And a couple of scenarios why one's oxygen might be low is just physically decreased red blood cell count. Okay, so maybe you're the person, right, who regularly goes to the Oklahoma blood OBI, right? Can you donate a Red Cross wherever? Okay. So you're always losing a lot of blood, right, effectively, as your body's triggering naturally erythropoietin to initiate this process. Maybe for some reason you move to an area where relative oxygen content is low. Okay, so like here in America, famously where Colorado, right? Go up in the mountains, oxygen pressure drops, atmospheric pressure drops, oxygen content is less readily available. So those people up there effectively will have more red blood cell count. Okay, their body will acclimate to that. Okay. Maybe your body just becomes more demanding for oxygen. So think about like pregnancy, or maybe you're exercising more, or whatever the case may be. Right? There's many reasons why your body might undergo the erythropoiesis process. Okay? And so if you increase erythropoiesis, the effect is to increase red blood cell count, which we know effectively increases your ability to carry oxygen. Hopefully at set point levels. Are you okay on the process of erythropoiesis, or the hormone called erythropoietin? Erythropoietin actually comes from a secondary endocrine gland called the kidney. Remember we talked about primary versus secondary? The kidney can release something called erythropoietin, and in this image it's illustrated in the green spheres. Okay, so if our oxygen content, remember our kidneys filter our blood, so our blood's continuously going through the kidney, and so it has the ability to monitor the blood flow, oxygen carrying capacity. So if over a period of time our relative blood count goes down, the kidney can detect this and try to offset it. It releases erythropoietin. The target is clearly which cells? Cells of your bone marrow, okay? And then trigger erythropoiesis, and then we make more blood cells. Okay? The kidney will determine that turn off, so negative feedback turns off the mechanism. This is summarized for you as well in the image, I think, coming up in your handout. So our red blood cells last about 100, maybe 110, 120 days maybe. And what happens is the red blood cells, as I mentioned earlier, can't kind of maintain themselves. They can't fix their proteins. They can't replace anything. And so they can kind of get caught at the spleen. That's one of the primary job of the spleen is to filter out red blood cells. And then it's got a lot of white blood cells in there, macrophages, and they're ready to kind of just catabolize or engulf, phagocytose your red blood cells and decay them, and then reuse what can be reused and just get rid of what doesn't need to stay in the body. So if we look at within the red blood cells, in the hemoglobin component, the iron core can actually kind of be salvaged a little bit. The protein component, the globin, right, it's proteins that can be broken down into what? Amino acids, okay, and they go in the blood and whatever, okay, so they can be reused as well. Okay. This overall life cycle of a red blood cell in your textbook is summarized in 15.4. You have an analogous one if you're on page 4 of your chapter 15 guided notes. At the spleen, an aged red blood cell is catabolized. The hemoglobin, we said, is broken down into, right, the globin part is broken down to amino acids. They could go to your skeletal muscle, or whatever, okay, they can be used, okay. The heme group is broken into the iron and something called bilirubin. You've probably heard of that before, okay. And at the liver, that bilirubin can be further catabolized into iron and into something called bile. You've probably heard of that too. You heard of that one? Bile? Okay. Bile can then be secreted and where's it go? Out. It is a nice way to say that. Okay. It's the bile which gives your output the characteristic colors. Okay. And so that color is due to the presence of bile. If the color of your output is like terribly dark, maybe some blood in the system, or maybe you're supplementing with iron or something like that. But if it's not dark like that, you might have, I said it's like white or something like that, you might have a blood deficiency. So that normal color is good. It tells you your blood cells are doing their thing and they're being cycled through. Yes?
I have kind of a little weird thing with that. My little boy had a tumor on his liver.
Oh no.
That we knew because his, when he used menstruum, it was white.
White.
So we went to the doctor because the bile wasn't getting into his feces.
Okay, so he's making the bile,
Yeah.
but it just wasn't, it was physically blocked and not able to. Okay, so interesting. So
we didn't know otherwise.
yeah, so yeah, it's a good indicator of health in general. It's not just being good. Thank you. I think that actually concludes our chapter 15 discussion. Okay. We'll conclude our discussion here, and I'll go over your exams, which I returned yesterday. Before I do that, though, to remind you, we're going to cover the respiratory system and the renal system in 3 days. Okay? So it's imperative that you do your reading over the weekend. Right? If you don't, it's just you're going to pay for it at some point in time. And I'd rather you pay for it this weekend getting ahead, and instead of when the semester's over and gone. So listen, right? So 16 and 17, go to the reading pages. We can't obviously talk about everything. Same thing, 18, 19, renal system. Okay, go to the reading pages. Go to the homework, watch the videos, redo the homework, rewatch the videos, redo the handouts to get ahead.